Dr. Ramesh Sagili is an associate professor in the Department of Horticulture at Oregon State University, and that's how we first heard of him, through the Pacific Northwest beekeepers. He obtained his PhD in entomology from Texas A&M University in 2007, specializing in honeybee research. His primary focus is on honeybee health, nutrition, and pollination. And Ramesh initiated the creation of Oregon Master Beekeeper Program and chaired the Oregon Governor's Task Force on pollinator health. His research program addresses both basic and applied questions to improve honeybee health and nutrition, and hence the majority of his research projects are collaborative, involving stakeholders. So I'm imagining you're going to share some of that with us. He has
Absolutely.
authored several important research and extension publications and has received several awards, including the Entomological Society of America's Pacific Branch Research Award. That was a mouthful. And the Eastern Apicultural Society's Outstanding Research Award and the OSU Outreach and Engagement Award. So we're very pleased to have award-winning Dr. Ramesh Sagili. So welcome, Ramesh, and get started as you wish.
Okay. Thank you very much, Steve. Let me share the slides and hope. Are you able to see the presentation view of my slides?
Yes, we are.
Perfect. Yeah. Thanks again, Steve, and Kathy and Bob as well, and all the organizers. I know it's been a phenomenon. I didn't see any glitches, and I knocked the wood here. So, yeah, this is one of the smoothest ones I have recently seen organized by beekeepers. So, looks like you are all proficient in Zoom operations. So, yeah, as I said to Steve before, Zoom format, I think many beekeepers are liking it. I know, of course, you can't have all the interactions, the personal interactions, which many beekeepers miss, but at least this has been COVID has been a blessing in disguise, that at least the Zoom format has helped us all learn and come together as researchers as well as beekeepers. So and again, as Keith said, this is my first presentation probably to your group. I don't remember or recall talking to New Mexico Beekeepers Association. And again, thanks to all of you, those who are here today. So I'll try to present some of our work that we have been doing over the last 6 years, I would say, on nutrition. And then I'll go over some basics as well. I imagine there are probably several of you that are very new to bees, and probably you just started last year, or I know with COVID, it has been a pain. You probably didn't have that much interactions, personal interactions to learn hands-on. And again, those who are planning this spring as well. So hopefully some of these might be useful as you go into your beekeeping. And here is my email address on the bottom of this. So if you have any questions, I know you may have a lot of questions, but if I'm not able to answer today, you can always email me at that address, ramesh.sagili@oregonstate.edu, and I'll be able to respond as soon as I can. So before I even start, this is a slide I always put to our beekeepers in all talks. All these exemplary people that have helped me run the program for the last, so I've been at Oregon State University about 10 plus years now. And before that, I was at Texas A&M as a postdoc, and I did my PhD as well at your neighboring state, Texas A&M University. So all these people, again, some of them are still around. Some of them have either students have graduated or some other technicians have taken other positions. But without this wonderful group of people, it's not possible to really do anything that we do here at Oregon State University. So I know you might have seen this type of slides a lot before, but again, just for the sake of the new beekeepers, I thought I would put this again just to remind them beekeeping is challenging. And again, I'm not discouraging. I really am glad that you are into bees and you're curious and you're wanting to keep bees. But again, be mindful, you will be encountering a lot of challenges here. And I have listed many of them that came to my mind. But again, I'm just going to talk about poor nutrition today, or at least address some of the nutrition issues in bees. And you heard an excellent talk by Randy this morning where he gave you a lot of excellent information on mite management. So those are still, I think at some point he said both honeybee nutrition and mite management are the critical ones that will define your beekeeping, whether you'll be successful in having a good survival after the winter. So again, so I'll talk about some of those aspects that I can. So again, nutrition is a big factor. as you all know, and this is one prime area of research. So we do a lot of work on bee nutrition and some pollination work as well, and varroa mites as well too. But nutrition has been a prime focus, at least for the last 4 or 5 years in my lab. And I know some of you had some questions. I heard about nutrition, how important it is in terms of managing mites or any other diseases. So I'll go over a little bit on that as well. So again, as I say always, nutrition is the first line of defense. Uh, I know there are so many things that are beyond your control, like your bees may get exposed to pesticides, or they may have some drifting going on, the mites may come in. So at least with the nutrition, I know you have at least to some degree some control. I know some of you may be living in areas where there is no pollen dearth. Maybe around the year you have good source of pollen, but there are locations, especially in the country, anywhere you go, there where you don't have a good pollen coming in during certain times of the year. So I think nutrition is very critical. I'll talk about carbohydrates as well. I know my talk is more biased towards pollen today, but bear with me. I'm not discounting the importance of nectar or honey that comes in. So the last line here I have is optimal nutrition has been shown. There are several studies, I think Kathy or someone was saying before, citing some studies from other labs as well. So there is enough evidence now that optimal nutrition or ideal nutrition, whatever you want to say, really boosts the immune system and decreases the susceptibility to pests and pathogens. So you can include anything from your mites to brood diseases or nosema, which is a gut parasite. And also the optimal nutrition also increases the detoxifying enzymes. So there is this P450 mechanism in insects and many other organisms where these detoxifying enzymes are very critical for detoxifying. Like suppose if your bees have good nutrition, and if they have been exposed to some small amount of pesticides, probably these detoxifying enzymes will play a role and help that bee survive. So nutrition again adds up to a lot of different things. So nutrition should not be ignored. So I'm trying to make bee nutrition, which is a complex topic, into a very simple topic here. I apologize if there are experts in the audience that know more than me here on bee nutrition. So I'm trying to simplify this for beekeepers that are new in the audience. So basically, when we talk about nutrition, you can classify that. I hope you are able to see my cursor. So macronutrients on the left of your screen and micronutrients on the right. So macronutrients basically are your carbohydrates, right? Your bees need energy to fly and come back to the hive. That's where your nectar and honey are providing them. And then we have protein, right? So protein, the pollen is the only source. So for those new beekeepers, remember protein is just coming from pollen. They are not able to get from anywhere else. So pollen is very critical, not for just for the uh, raising of brood, but also for the colony growth in general. And there are protein needs for adults as well. So then micronutrients is an area. And before I go into micronutrients, I just have to emphasize that bees have been studied well over 200 years, at least very meticulously. But if you look into the literature, bee nutrition is one area that has a huge gap in knowledge. So that's another reason why many labs have started looking into honeybee nutrition, including ours. So if you look into literature, you'll find some information. I'm not saying we don't know anything. We do know, but again, it's very basic. So we have barely scratched the surface. So especially there is lack of information regarding micronutrients. So when I say micronutrients, these are required in small amounts, right? So they could be vitamins, they could be minerals and lipids. And lipids, again, I'll go a little bit over lipids in the next slides, but today we'll be mostly focusing on sterols. So sterols are a type of lipids as well, right? So let's talk a little bit about pollen. As I said before, pollen is the protein source for bees. And when we talk about protein, that it's mostly about crude protein in the pollen, and also amino acids. These are two important ones when we talk about pollen. And most pollens have been shown to have about 10 to 40% crude protein. So that's a range. So remember, all pollens are not equal. Like you can't compare one pollen to the other literally. No, they're very different in terms of what amino acids are present or absent, the crude protein, the sterols, the phytochemicals. There are so many things that are different. But this is the crude protein I am talking about here. They could range anywhere between 10 to 40%. And as I said before, there also that means pollen. Pollen is a source of lipids. Again, sterols are more important ones in that, and I'll go that over that in a minute. Minerals and vitamins. Then nutrition, I know it's important throughout the year, of course, but again, if you want me to just pick one point of time when I think is the most important, I would say it's during the time when your bees are raised, the winter bees are raised. Again, it might be very different in different locations. So I am in Oregon, so our weather is temperate weather, whereas yours is more subtropical, arid, dry weather. So it might be very different in the life cycle of the bee as well in your area. So nutrition, again, just remember whenever you think, or at least you can do an experiment. I have a quick study that I'll show you on diatomaceous bees. Hopefully I'll have time to go over into that one as well. So the nutrition is crucial when they're raising those winter bees, because these are the bees that are survived, and now what you see in your hives today, they were the ones that survived the winter, however harsh your winter is. I know you don't have a bad winter weather, it's pretty mild probably, but still your bees will be the ones that are raised late fall or close to the start of your winter time, is the time when you get your diatomaceous bees. So diatomaceous is just a Latin word. Diatomaceous means long-lived or long-lasting. So these are the bees that survive almost like in a temperate weather like Oregon, or if you go to Minnesota, where there is almost a 6-month winter. So those bees that survive are almost 5-6 months old when they come out to forage again in spring. So those are the winter bees. So they are raised basically before the brood rearing season stops in that area, right? So that's the time when you want your bees to be, as Randy mentioned, both varroa and nutrition are critical. You have to manage your mites so that you have less transmission of viruses, and you have to have your bees as fit as possible in terms of nutrition. Probably that will define your success for that year. So this is just to make you think about what challenges you may have in your area in terms of nutrition. Again, this might be more valid to commercial beekeepers. I know most of you are backyard beekeepers in the audience. So think about it. So lack of pollen abundance is one thing, right? When I say abundance, it's basically quantity. How much pollen is coming in a month or in a week when you need that brood rearing is going on. So that's important. You need to know whether you have good quantities coming in for the hive to thrive. Then there is lack of pollen diversity. I think some of you were asking or talking about this before, about the lack of pollen diversity. So pollen diversity is basically what types of pollen, right? What different types of resources you have. So that's important. Like, are you getting like 5 different types of pollen coming into the hive, or or is it just one source coming in? So that's basically diversity. There are a couple of studies I have seen, but this pollen diversity area has not been explored very well in many studies at this time. So, but at least a couple of studies have shown it's important to have pollen diversity. So lack of pollen quality is another one, where you're looking at this quality of the pollen itself. So when I say quality, again, it's a very broad, ambiguous term. So at least to make a little sense for you, I would say protein content, or the lipid content in that pollen, including your sterols, phytochemicals, those all different things. So that define a quality of the pollen. So lack of pollen quality is important as well. So for example, we have some hybrid carrot seed production here in Oregon, and because it's a hybrid crop, it doesn't produce enough pollen, and also the quality, when you analyze the pollen, the crude protein is pretty low. So that's what I'm talking about. So look for those things as well. The pollen that is coming in, is that of good quality as well? So like, and it's assumed that most of the wind pollinated species have a low pollen crude protein content. It may be true sometimes, it may not be. I have seen some wind pollen like hazelnut. We have a lot of wild hazelnut and cultivated hazelnut here in Oregon, which is called filbert. I don't know if you have heard of that before. So that's I used to think it's very low quality, but recently we analyzed that pollen, and it's pretty close to tree fruits. It's about 19% crude protein, and many of the tree fruits like your apple, pear, cherries, they end up around 22-23% crude protein. So now I'll dive into some of the studies that we have done in the last few years. Again, I'll not bore you to death with a lot of details here, but just to give you some sense, and maybe some of these results you can relate to your experiences as well. So this is the first one where we looked at pollen diversity available to honeybees in 5 major cropping systems. I'll show you those crops in a second. So basically, we were trying to see what kind of diversity do these bees have access to in terms of pollen when they are in all these different cropping system, because an Oregon beekeeper on average pollinates about 5 different crops. So here again, I apologize if you can't read well, but I'm hopeful you're looking at your computer screen, so it would be easy for you. So if you look on this far left, these are the 5 different crops: almond, blueberry, carrot, cherry, and meadowfoam. For those who don't know meadowfoam, it's an interesting crop. It's an oilseed crop. It's only grown in parts of California probably, and Oregon. Um, very, very beautiful, and it's good source of pollen for bees. So if you look here in this, where my cursor is, if you can't see, it's a third column I'm talking about, where it says mean number of plant taxa. So here you can see when bees were in almond, they got only 3 other different species of pollen. So its majority was almonds. And blueberries, again, they're notorious here in the sense blueberry is not a great crop for bees to be in because of the flower structure. Bees don't collect enough pollen. So bumblebees do a great job of pollinating blueberries, but honeybees do pollinate those crops. Mostly the nectar foragers probably are doing the job instead of pollen foragers in blueberry pollination. So you can see there is a huge range of plant taxa found in the pollen that was collected in hives from blueberry pollination. And carrot also same, there is about 11. So this tells you that it's not always same. You can't expect put your bees in a certain cropping system or a certain location and expect that uh, you'll get a lot of diversity, or sometimes you may get, sometimes you may not. So there is a huge variation. That's what I wanted to show you here. And here you can see this is in grams per week. So in almonds, about 300 grams. Blueberries didn't give much, even though there was diversity, but total volume of pollen coming in, or the quantity coming in, was not enough. And same cherries was fine, and meadowfoam got a significant amount of pollen. So for those of you who are confused why I'm showing in grams, unfortunately, we publish in a metric system. So here is uh, in pounds, so that you can relate to what was coming in a period of 1 week in these crops. Yeah, so lipids, this is what I said I'll focus more on, and especially, so lipids broadly, you can classify them as fats. Probably you heard of triglycerides, then phospholipids, steroids, which include sterols, and then waxes, like our bees also produce wax, so that's probably a lipid as well, to have an example. So today I'm gonna focus more on sterols because that's where we have done our studies in the last 3 or 4 years. So a little bit introduction for sterols. So sterols in insects, they are components of cell membrane, and they're also precursors. That means without sterols, you can't have molting hormones. And molting hormones are very critical, right? If you think of insects, if molting hormones aren't there, then you are stuck in the same phase that you are. If you're an egg, you'll be an egg. If you're 1-day-old larva, you are a 1-day-old larva. You don't go to the next stage. So molting hormones are very important for insects to get to the next stage. So sterols, that for that molting hormones to exist, you need sterols. And I think I have a video to show you later on that as well. So sterols are important for us as well, right? So I know I can't see you guys, so I'll just make you think. So what is your sterol? What is the important sterol that you need as a person? So you probably are thinking of cholesterol. So cholesterol is our major sterol, and cholesterol, now we hear that cholesterol is bad. Cholesterol is bad only if it blocks your arteries, if there is too much, but you need cholesterol for all those different functions I mentioned here in insects, as well as many other functions that we have as humans. So sterols are very important. And so for honeybees, it's not the cholesterol that we use. They have a very specific cholesterol, 24-methylene cholesterol. I know it's a mouthful. I'll keep talking about this because I have 2 more studies to show you on this 24-methylene cholesterol. So for now, just remember it's a major sterol source and it's obtained from pollen. So anyhow, there is no quiz at the end, so you don't have to remember this name. And I don't know if I said this, but all insects, including honeybees, they have to get their sterol from the diet. They can't make their own. So for us, our liver helps with the sterol, but with bees and other insects, it has to come from their diet, and their diet that provides them the sterol is the pollen. So when they have this, so this is general for insects, not for honeybees here. So the sterols may be in different form. There is cytosterol, there is stigmasterol. These are all plant sterols. So if an insect takes these sterols, it has to be eventually metabolized into some form of cholesterol. In our case, with honeybees, we need 24 methylene cholesterol. So a little bit about that molting hormone that I mentioned before. So here you see your larvae, right? There is an egg. There is probably a less than 1 day old larva here in the brood food. So they are all molting and then getting to different stages. So these are two hormones. There is ecdysone and juvenile hormone. So juvenile itself says young, right? So this ecdysone and juvenile hormone, they have this interplay going on. So they have to balance each other. So if you have high amount of juvenile hormone at some point, then you stay in that stage. So if this larva is a 1 day old, then it will stay in that 1 day old until this ecdysone levels go up. When this goes up, then it will mold to the second stage. So that's how it works. It's basically, I'm oversimplifying the molding hormones here, but you don't need to know that much for as a beekeeper. At least understanding that sterols are important for molding hormones is good enough. So there is something you might read in literature if you are getting more interested in trying to understand more of the sterol. So there is endogenous sterols. Endogenous sterols means sterols that are there incorporated into the body. So when a nurse bee consumes the pollen, she's getting that stores of sterols in the body. So that's what I mean by endogenous sterols. So there was a study in 2008 I saw. They found that newly hatched larvae that lack dietary sterol, they usually die in the first or second instar, and because the sterol is exhausted. So that's what they are getting it something from the egg stage, but after that, if they're not being provided in terms of diet, then probably they're not getting to the next stage. And that's what I said before. And if you didn't get anything and after lunch you're sleepy, just watch this animation. Hopefully what I said is encapsulated in this animation. So here is your pollen. So those pink dots, I'm just depicting them for sterol. So these bees come in and they feed on the pollen. Hopefully these are nurse bees. And this is what I was saying, the endogenous sterols. So they get into incorporated, they will not see them outside. I'm just saying that they are incorporated into their fat bodies or the hypopharyngeal glands, wherever. So here is a bee that has developed because the sterol was there. But again, just an imagining another scenario where you have made your own protein supplement or you bought from a store, and if they are lacking in sterols, so you're not seeing any pink dots, which are sterols. And then you have the bee, which didn't have much sterols in her body as endogenous sterols. And so this larva never got any sterol. So it just desiccated or didn't get to the next stage. And then, so there'll be no molding hormones, right? Because there is no sterol. And that's how your colony may not grow. And then the bees may just cannibalize this larva if they feel that this larva isn't growing. I hope the video was working. Steve, can you tell me if the videos are working? Did you see the animation?
Yeah, it was great. I was trying to imagine which one of your postdocs took the time to put that all together for you.
Yeah, we'll tell that secret at the end.
Okay, great. It was very good. Thank you.
Yeah, yeah.
It played very well.
Okay, thanks. Yeah, sometimes with the Zoom, it's yeah, I'm glad it played. So the next slide that I'm showing you here is we looked at this 24 methylene cholesterol. That's of our interest, right? So we looked in one of those commercial diets available, and also looked at in almond pollen. So you can see in almond pollen, we could easily find a decent amount. It's in milligrams per 100 grams here. So again, just to show that all pollens will have, but it might vary to some degree. And I know some beekeepers use oils. They mix maybe canola oil or some type of oil uh, to provide sterols. And so here I'm just showing some oils here. It's a borage. In this study, at least, you can look at this study. I think it might be available for you to look at. So 22.9. So borage is the only one I could find among the categories of oils where you could see a, a significant amount of 24 methylene cholesterol. So now I'll go into some studies that we did with 24-methylene cholesterol. So basically our goal here was to evaluate the dietary sterol, because if you look at the literature, there are studies done in 90s and probably late 80s as well, on 24-methylene cholesterol as well. But it's not very clear from any of those studies is what is the exact amount we need. What's the ideal amount of methylene cholesterol that we need for optimal growth of a colony or optimal development of a larva, right? So that was one goal. And the other one was we are interested in looking at sterol composition of the forage. That forage, again, when I say forage, it might be misleading. What I mean is the pollen forage that bees have access to. So we want to look at several pollens that are common, or at least bees use them for their colony purposes. So we want to look at sterol compositions on those too. So I'll show you a couple of studies here. So again, I think this might be an open access as well. If you're not able to access this study, let me know. I can send you a PDF of this. So here what we did is we wanted to evaluate the critical micronutrient, which is 24-methylene cholesterol, on honeybee physiology. So we wanted to look at the physiology, also look at what is the ideal concentration we need. So this is a cage experiment. As you see, we have these cages in the lab where we bring in newly emerged bees. We bring frames and immerse the bees in the lab, and then those bees go into these cages, and then we provide them with water, sugar, and also with the methylene cholesterol that we were supposed to provide them as well. So we had these treatments control which didn't get any 24-methylene cholesterol. Then we had these increasing doses of sterol diets from 0.1, 0.25, 0.5, 0.75, and 1%. And then I should quickly tell you the story of 24-methylene cholesterol as well. It's a very expensive chemical. So some of you may say, oh, can we not just use pollen? Yeah, of course. As a beekeeper, you will use pollen to get some 24-methylene cholesterol to your bees. But for these studies to tease apart all the confounding variables or the background noise, you need to have an exact amount of just 24-methylene cholesterol, no other background noise creating like any other sterols in the pollen that all creates problems. So we had to look for a pure source of 24-methylene cholesterol. And when we did this study about 3 years ago, we didn't find any source. Probably some of you that have done some research in the past, Sigma is a company that provides reagents and a lot of chemicals, and we couldn't find this in any of those catalogs. So eventually we asked a Canadian company that had in their catalogs, but they didn't have stock. So they had to custom synthesize this 24-methylene cholesterol, and it's pretty expensive. You'll be surprised if I tell you the price. It was about $80,000 for just 10 grams. So again, Priya, who had done this work, her hands would shake when she held the vial. If you drop the vial, $80,000 is on the floor. So we were able to successfully finish this study. But again, I'm just saying, those who might be looking for 24-methylene cholesterol in the market, there is nothing that is sold. This was something we got it from a lab that custom synthesized for our research. I'll quickly go over the results here for this sort of diet consumption. So here you see week 1, week 2, week 3. That's how we did sample, or at least the data collection in those cages. So you can see here the consumption for the higher concentration. So here, this is the highest concentration. I don't know if you can see the cursor, but the B, the one on the week 1, the last bar you see, the dark one, that's the highest concentration, which is S5, which is 1% sterol. So you can see they were not significantly different. Those letters mean that they're not different from each other. So those all B's means they're all similar, not statistically different. You may see some differences visually here, but they're not statistically different. So it looks like all the higher concentration ones, the consumption was high. So that tells us that the bees were able to detect the presence of sterol in those diets, and they were selectively consuming higher quantities of those when compared to the control. So your bar here, the light one without any shade or dots, that's the control. Here on week 2 also, that's the control. So we looked at the mean head protein. When I say mean head protein, you probably heard of those hypopharyngeal glands. These are the glands in the honeybee. Nurse bees have these very well-developed glands. After eating pollen, they grow those glands which produce the brood food. And these are called exocrine glands. So exocrine means they are letting the thing out, right? They are opening outside. They're not just getting into your bloodstream, but they're opening outside through your mouth. So you have these hypopharyngeal glands and mandibular glands in the head, which are the brood food producing glands. So that's the reason why we look at the mean head protein. So you can see with the sterol, highest diet sterol, you had a high mean head protein as well. And then with the mean abdominal lipid, again, we can see here the abdominal lipid, we didn't see a difference between at least the highest ones. This probably is 1, 0.75, 0.5, and 0.25. They didn't differ very much, but they all are significantly higher than your control. And then we also looked at survival after providing these different concentrations of sterol. So I apologize if you can't see this very well, but there is an orange bar on the top, which represents this S3, which is your 0.5% sterol diet. So that one was the highest, that one had the highest survival, not the one with the green S5. So that's why is somewhere there on the bottom. You just have to believe me because I don't think this is very clear on the graph here. So basically what we can say from this is looks like you don't need a 1% diet. You probably 0.5% diet is 0.5% sterol diet is still giving you the highest survival. So we also plan to do some, we haven't done, we did some preliminary work, but we also have plans to do some electrophysiology work. Basically what we want to look at is we plan to examine the impacts of nutrition on memory and learning, because that's another type of behavior that might be impacted. And so we haven't started this yet, but we did have some very preliminary information. I'm not going to show that today, but so here you can see this antennal lobe. This is a mushroom body. Mushroom bodies, they're a center for learning and memory in honeybees. So we are inserting a probe into this. These are the electrodes. If you have ever read anything about electrophysiology in insects or other organisms, so it's basically an electrode goes and wherever you want to record the response. So here this probe is getting to the brain of the bee and getting to those mushroom bodies, which are responsible for learning and memory. And we do the recording. So all the recordings are happening on the computer, which I don't show you here in this slide. I think, yeah, this video you can see, this is a live bee. The top portion of the head has been removed, and now you can see the brain, and that's a mushroom body. So this probe gets, and then we are reading the responses on a computer screen on the other side. So again, this is something ongoing. We don't have much data on this one yet, but that's another plan to look on to these olfactory cues or memory and learning if it's impacted because of nutrition. So take home message, as I said before, the bees that received the highest concentrations, they were able to consume more, and that tells us that they can perceive the sterol in the diet, and they are consuming that in a higher proportion to meet their needs. And then as I showed you in that survival graph, the 0.5% concentration of 20. So when I say 0.5%, it's the 0.5% of the total weight of the diet that you are giving, right? It's a small percentage. And then the honeybee protein the head protein content was higher in bees that consumed diets with 24 methylene cholesterol. So again, lab studies can sometimes may not be that insightful. So you may have to expand the study to the field, and that's how many of the researchers do. So we expanded this study into the field, which is, again, in the field, you can't openly do this because again, there will be so many confounding variables. So to control most of those variables, it has to be a flight cage study like you see here. So what you're seeing, it's a drone image from the top. So each of those square-like things you're seeing is a flight cage, probably 12 feet tall and 6 feet wide. And there is a small nucleus hive in each of those. I don't know if you can see, but that's one nucleus hive. That's one. So we have completed this study, but unfortunately we haven't analyzed the entire data. So I'm not going to show much of this as well. Maybe next time at some conference, you may hear me talk about that study too. So we also have this, as I said before, we also want to analyze these different pollens that are available to bees, as many as we can, and look into sterols and a lot of other nutrients as well. So this was another study where my postdoc Priya was just looking at the sterols in some of the pollens that we wanted, and also some of the protein diets and some oils too. So this is again an open access journal article, so you can always just Google for those titles and find it. But if you can, just let me know. I can always provide you that as well. So in this one, what we looked at was sterol profiles for 5 commercially available crop pollens, and then 3 corbicular pollens. When I see, for those new beekeepers, don't get confused. When I say corbicular, it's nothing a magical term. It's the pollen basket is called corbicular. So bees collect the pollen, honeybees collect their pollen on their corbicula, right? So that's what we use the term corbicular pollen. And then there were 3 vegetable oils that we tested, and there was 1 commercial protein supplement as well. And yeah, we were fortunate recently, just like a month ago, we received a grant from USD and NIFA where we proposed to look at to develop a pollen nutritional composition database. So it would be a very comprehensive database looking at sterols, protein levels, phytochemicals, amino acids, a lot of other different things that we can. For at least 100 species. That's a little ambitious, but we'll see. It's a 3-year grant, so hopefully we'll be able to achieve those targets, and that might be a good information for people, because there is a lot of news about, you read, and maybe some of you may ask me at the end of the talk, give me 5 different plants that are good for bees. I will be ready for that, but we'll see. So you see this Jersey Society or Project APSM, many of them have a lot of excellent information on plants that you can plant for your bees to provide both pollen and nectar. But again, the limitation at this point is most of these plant recommendations, they come from attractiveness, right? So if something is attractive, doesn't mean always that it is a robust or it's an optimal plant. Maybe if you have some lavender around you and nothing else is in bloom, so bees are just going to be crazily foraging on that. That doesn't mean that lavender is the best source, right? It may provide a lot of nectar and some pollen as well, but then no one has tested there is fat bees, skinny bees, and even I think Randy has a nice at least a couple of articles on nutrition. So again, if you see that all the synthesis of information, but none of the studies have comprehensively looked at all different things. Either they have done just the protein for several pollens, or just amino acids, or some sterol. So here our goal is to have a more comprehensive database that can help people to choose some of the plants, or at least those who are recommending for planting to develop habitat. It might be useful. So we'll see. I know it's an ambitious project, but I'm excited that we finally got some funding from the USDA to do that. So I'll quickly go over some of these slides here just to show you what we are doing. None of these might be really useful at this time, except for 24 methylene cholesterol. I'll stop on that slide. So you can see here we looked at borage. This is an oil, canola oil, soybean here. This is the commercial diet, CD. FP means Ferment Pollen. So Ferment is a company somewhere in Washington that sells hand collected pollen. So we wanted to test those as well to see if they have very much different, are they very different when compared to what bees are collecting. So Ferment Pollen, there is a Ferment Almond Pollen, this is apricot, this is apple, I guess, and this is cherry, this might be blueberry. So we have all this. These are California collected ones, or sorry, the corbicular ones that bees have collected. So you can see there is a lot of variation in that, but even commercial diet, some sterols are there. Like here you can see there is cholesterol in here. There is a beta cytosterol there. So let's go to 24 methylene cholesterol. So this is on your right side is the 24 methylene cholesterol that is of our interest, because that's a major sterol that our honeybees are looking for. So you can see most of these pollens, so again, the same letters D means they're not significantly different. So they have a lot of similar amounts of 24 methylene cholesterol in most of these pollens that were bee collected or collected directly from the plants. Uh, so this is apricot, that's apple. So, and you can see this is the commercial diet here. So you see some small concentration, but it's pretty low when compared to 24 methylene cholesterol that is present in the diets that had pollen. So again, desmosterol, ergosterol, these are different sterols. I'm just showing that we have the ability to do all these different sterols, plant sterols, to get analyzed in different pollens or diets that are available. So I'll stop that talk there and go into diutinous bees. This is probably more uh, interesting for some of you that are curious about these winter bees. So as I said, diutinous bees are winter bees or fat bees. Some people use those terms. So the lifespan of these bees is longer, as I said before, right? So I just want you to think why they have a longer lifespan, because they have same genetics, right? They are progeny of the same queen, but then why would they survive more in winter than in summer or spring, right? So these are okay. I'll get into that soon, but just let's go into the physiological differences uh, between those two bees, the ones that are raised in winter versus those are in spring or summer. So the winter bees have large intrinsic protein stores. They have high fat body content. I think I'll show you that image. I took this from Randy's website because I couldn't find the other original one from that study. So yeah, probably if you have been to Randy's site, you might have seen this before. So here is a picture without any much of the fat bodies. This is a forager, but here is a nurse bee. You see how much dense fat body that you can see? This is the abdomen that you're looking at. So again, probably if you're very curious, have good microscopes at home and you want to dissect or collect some bees in spring and take some images, and same colony you can do in winter, get some bees and see, probably you will notice a significant difference in their fat body content. And then I think Robin or Margarita mentioned vitellogenin, which is a glycolipoprotein, which is very critical one for their longevity. So you will see if you do an analysis for vitellogenin, there is a high amount of vitellogenin in those winter bees. So these are the differences basically. But then I just want you to think, why would they have these differences? Why would they have this much protein and fat bodies and vitellogenin? And that's the reason why they survive longer. So I want you to think what happens in spring or summer. So suppose you are in June and you have a lot of brood. Maybe you have 6-7 frames of brood, and there are nurse bees, right? There will be always nurse bees in the colony. Uh, so so they are feeding, probably, just to make an example simple, you have 8 frames of brood to feed and you have certain number of workers, and then suppose there are 1,000 nurses that are feeding. I'm just making up some examples, there will be a lot more. So these 1,000 nurses were feeding 8 frames of larvae, and they were continuously eating the protein, right? There is a lot of pollen coming in, they're growing these glands, feeding the protein to these larvae, and so they're not storing anything. If I am I am a nurse bee in spring or summer, I'm not storing anything in my body much. I'm just barely meeting my requirements, my physiological needs. But most of the stuff I'm eating, the pollen, I'm converting it into the brood food and spitting that into those cells where the larvae are, right? So larvae are being progressively fed with that protein. So I'm not storing, I'm not selfish. Again, I hate to use this term, but I'll say this bee is not being selfish. She's just providing everything to the larva. But what happens in September or October, whenever your winter is going to set, so you have less larva, right? Now instead of 8 frames, there is only probably a frame or a half a frame, whatever, in your area, and you have still a decent number of those nurse bees. But now they are still eating pollen if it's coming and it's available, but they're not feeding the larva that much, because there's only a few larvae left in the hive. So they're storing everything that they are eating now, and they're creating those high reserves of fat bodies, and that's why they survive longer. So that's a physiological difference you should think about. So I used to get this question always. I'm getting close to my time. I think I still have 15 minutes. Okay. So when are these winter bees raised, right? So that's a question that probably some of you already are thinking about. So again, this study may not be very useful for your area, but we did a quick study here, at least 2 years, for our beekeepers in the valley. So we are in the Willamette Valley, which is on I-5. So again, Oregon is very diverse as well. We have these Cascade Mountains, there is coastal range, and so it may not even pertain, but at least in the valley, we uh, think this might be the uh, uh, a phenomena that is going on. So what we did is we obtained this again in the earlier experiment I mentioned. We bring these newly emerging bees means the bees that are ready to be emerged. So we put them in incubator at the same hive type temperatures, 33 Celsius, about 50% humidity, and then once they hatch, then we paint them with colors for for every 2 weeks, there will be a different color. Like in this image, you may see some colors. There is green, red, white, yellow. So that's what we did. So we have this big bee painting fun exercise. I'm just saying fun, but it gets tiresome after some time. So we do all this for every 2 weeks. It gets a different color, and they all go into certain hives that we have designated as foster hives. So that's where they will age, or they will overwinter, and then we will go back in March and find out for each color how many bees were surviving. So that's what I have here for the 2017 study, where so we released bees in September. So that's what I say, bees emerged or born in September. So they were in 2016. So we go back in March in 2017 and find out if I released 200, how many are surviving? So that's the percentage that you're seeing on your right. So the 35% of the bees that were raised during this time, they were still in the hives when we opened those foster hives on March 14th, 2017. So for the ones that were 2 weeks later, about 28%. So you'll see some variation as well. And again, these are not perfect studies where if you replicate this few times, you may have some variation. But again, I'll show you which month was a little more consistent here. So in October, in our area, about 46%. This was the highest we saw. About 46% bees were surviving that were raised during this time. And then it decreased again a little bit when we get into later October. And then November, we had about 32%. So I'll show you one more year data. Here, this is from the next year, 2017-18. You can see October looks like a more promising time here. So again, this is not something really critical, important information, but I just want you to think about those bees, and you don't want any nutritional dearth or any nutritional deficiencies occur during the months when in your area those bees are raising. Again, October may be a wrong month for your area. You may have to do some of your own work in New Mexico to get some range or period when you think the bees are raised, and those are the ones that are surviving most of the time, or more percentage of those bees are surviving at the end of winter. So again, just to give you some insight so that it makes you think about your winter bees, which are very critical. So take home message, your adequate nutrition, especially protein, as I said, is critical during fall and late fall. So whatever that is in your area, I can't define that. So look for that when your bees are, that would be just before they either slow down or totally stop laying in your area, the queens. And then winter bees are at least very important. As I said, again, I'm just reiterating the same thing. Winter bees need utmost attention, both I was quoting Randy before, both nutrition and varroa mites. So when I say health, there are other things as well, but varroa mites are the most critical ones. And so you have to make sure that you have good control of varroa mites by the time your winter bees are reared in your hives, and they have optimal nutrition as well. So probably that might increase your overwintering success as well. So this image just I put to show some of you probably are curious about how to detect starvation, right? I know this is not always going to happen. So you may see here, this left image is full of larvae that are floating in the brood food. So you'll see a lot of brood food in these cells, the shiny glistening stuff. But here, if you look on the image on the right, the lower right, you see there is not much, right? Some of them have, even the larvae have moved out there. They usually don't move that much, but then because of the lack of this brood food, they may even start curling and trying to get out to see if they can find any food inside the cell here. So if you see some of these dry cells, if you are, again, unless you observe those frames very carefully, it's hard to say. So if you're meticulous observer and looking into this brood cells, and if you find something which doesn't have or is lacking the brood food, literally dry, so then it's a very good storytelling here that your colonies don't have adequate pollen in the hive, so they're not able to produce enough of those brood food using their hypopharyngeal glands and mandibular glands. These are some fun examples I have put here. So you might have, some of you probably have seen this. So on the top one, what you see here is these bees are collecting some corn flour or cracked corn pieces, small on their legs. So this is from one of our Central Oregon areas where there is yeah, we do a lot of work with the hybrid carrot seed producers in terms of pollination and honeybee health. So some people call me that there are crazy bees in these bins with cracked flour or cracked corn and some flour in it, and these were collecting on their legs, so they were thinking it's pollen. So that again tells that about the area. It basically tells you that this area where bees are doing this act, it's probably very poor nutrition. In terms of nutrition, it's not a very good place. So that's why they're desperate to find anything that looks like pollen. And we went back to the hives that were close by to this facility, and you'll see a lot of this stuff being thrown out. So because these are foragers that are collecting, but once that gets in, then there are bees that are packing and storing, right? So those bees recognize this is not pollen, and they'll start throwing it out. So again, if you see some of these scenarios, that tells that you are in an area which has nutritional dearth. So here is a bee collecting some rust spores of fungus. This bee is collecting some honeydew. And this was the most fun example I have. I don't know if you can see the contrast here. So you see these bees have some reddish abdomens, or whatever color you see, a little brownish, reddish. So these bees were collecting some grape juice. These are some of our Oregon State University hives. This was like probably 8-9 years ago. So unless these bees are on a white frame, I couldn't see, right? If they are on this wooden frame, probably the contrast isn't there. So we were curious what they were bringing. So we had to kill one bee and then take the gut off and taste it. And it was tasting grape, right? So there are some quick tests as well. As a beekeeper or a researcher, you can do some tasting as well to see what the resource is. Um, I think I'll wind up in 5 minutes with this supplemental forage study. So Steve, I have 5 more minutes, or
Oh, you're fine. Five
I'm
minutes
fine.
is good. We,
And then
we're gonna,
I have
we're,
questions time as well. Okay.
of course, yes, we
Okay.
will.
Perfect. So here we have been doing some studies with farmers as well in Central Oregon, where I mentioned that hybrid carrot seed crop, where beekeepers are not really happy when they go back home after a month of pollination, because most of this area is very ag intensive, especially the hybrid crop, where these bees are placed for pollination, doesn't produce enough pollen and nectar for bees. So we are looking for some alternate options. Like I think some of you were interested in, I think I saw some questions in the past asked about developing habitat for bees and all. I know it's a big push. There was this 2014 presidential memorandum during Obama's administration about improving habitat. So there is a lot of work being done across the country on improving. Even in almonds, there is a lot of push to have these almond growers adopt some of these practices where they can plant something for bees before pollination. That means before almonds bloom and after almond bloom is done. So here in Oregon, we have been testing some of these plants like Phacelia, mustard, borage, buckwheat, and I'll show you some more in the next slide. Um, so here you can see the others like bachelor's button, Alyssum, Alsea, clover. So we have been testing this for the last 3-4 years. The challenge is you may ask, what am I testing for here? So not just looking at the protein and the nectar volumes in these different species, but also to see if it matches with the time. And because we are doing this in keeping the commercial beekeepers in mind, right? So if a commercial beekeeper comes in July, first week of July is the time around Independence Day, around that's the time when uh, they come for pollinating uh, this hybrid crop. So we want this to bloom at the same time when the bees are there. We don't want it to, because if it blooms before the bees have come, then it's not useful either. And those bees don't stay too long after they are done. So we want this to happen at least mid bloom. So I know it's hard to convince producers. Probably you have interacted with farmers as well. It has to be a win-win, right? That's a challenge as a bee researcher for us, or for beekeepers as well, is to convince them what is in there for them. If they have to plant this additional acreage next to their farm or their orchard, wherever, it has to, if the first concern is, are those bees going to be distracted? Will they all go into this more uh, 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 lucrative looking or more honey producing type or pollen yielding variety, rather than getting into my hybrid carrot seed crop? So we had to come up with all those, and so we had to look for whether it should not become a weed if we suggest some plants, and if they become a weed and it becomes a weed control problem for them, irrigation. So there's so many parameters. That's the reason why we are doing this on a continuous 3 or 4 year scale, to see that we can avoid those and come up with 2 or 3 best species we can recommend that these growers can use. And we have come up with, at least the logic for them is, the brood pheromone is important, right? The larvae in the colony are the ones that are telling your pollen foragers or nectar foragers to bring the resources. So it's all tied up to, there is a feedback loop with the brood pheromone in there. The brood pheromone is being released by the larva. So they can communicate with the bees directly. So they're communicating with this chemical communication mode, which is the brood pheromone. So what we are telling these farmers is, if your hybrid carrot may be okay, they may be there for 10 days, the bees may be there because they're not finding anything else. But then what happens is the queen is slowing down as well. And we have documented that as well, because there is not much incoming pollen when they are in this pollination scenario. So there is less brood, and then there is no stimulus for bees to forage on their carrot seed crop as well. So I think that's the biology aspect we have to convince farmers, and we're not making up things here, but that's what it is. It's all directly related. So if you don't have enough brood in the hives because of lack of forage, then those colonies will not do a great job of pollination either. But anyhow, bees are always distracted. At least 20-30% may be going to some other resource, even if you don't have anything around, right? So those are some ways to convince farmers and have some acreage available for bees, which is a win-win thing. I think growers do agree, at least those who have agreed to plant, they are convinced that, yeah, it's a win-win scenario for them. We are not asking them to have acres and acres. We're just asking, if you have 10 acres, can you spend at least a quarter acre or a half acre in additional supplemental forage? So here is an example of the carrot crop. Here you see carrot, and here is the bachelor's button that some farmers planted around. Again, we haven't recommended anything yet, probably next year, but some have already tried something like this. This is in almonds. You see that's in the background. The almonds are not yet in bloom, but this farmer has planted rapini, a type of mustard. So again, what I want you to take home from this is, make sure your bees have access to adequate pollen throughout the brood rearing season. I know some of you probably live in very beautiful areas where there is no dearth throughout the year, but I know many of you probably face a pollen dearth around the year. So be careful when that time is, and use some protein supplements, and I would say supplements which have about 5 to 10% pollen, because with that sterol that I mentioned, you need those sterols, because many of these diets probably don't have adequate amount of sterol that you're looking for. So please add some pollen of your own, or ask for, when you're buying, something that already has 5 to 10% pollen mixed in. And I'll show you how to trap pollen in the next slide. That's my last slide for the day. So adequate nutrition is critical during fall and late fall when winter bees are raised. Again, I'm just reiterating the same thing. And winter bees are important, as I mentioned before. So this is my last slide, where I'll just give you some tips, because I have been asked in some other beekeepers conferences on if they can collect their own pollen. So yeah, you can always collect your own pollen, because that's a more safer mode, right? Because if you're buying pollen from unknown sources, most of them probably do a good job of irradiating the pollen, because there is a risk when you're using pollen from some source which you don't know, because there could be chalk brood issues. You could get some other brood disease problems as well, because the pollen may be contaminated. But if you know that your hives are pretty healthy and you want to collect your own pollen, then these are some tips I have here, very quick ones here. So the front the front porch traps are the ones on the top. These are simple plastic ones you can get from any bee supply store. um, I mean, there might be some wear and tear more than these type of traps on the bees, because these are a little more harsh on the bees. But again, you're not putting them for too long. So, and it's easy to collect. You have this tray that goes in here underneath this here, and this just sits in the front. So just follow the instructions, and I'll give you a good a manuscript as well, which you can read to be successful in pollen trapping. So these are front porch, these are bottom mount. So they go on the bottom of the hive. These are a little more complex to put on because you have to lift those boxes and put the pollen trap, and it takes bees a little time to orient here. This is relatively easy to get orientation for bees. But if you just need, I don't know, you as a backyard beekeeper, depending on how many hives you have, you're not looking for huge amounts of pollen to trap. So I'm looking for maybe even 2 or 3 teaspoons for each of the hive that you have. So it's not a huge amount that you're collecting here. So these are some tips here. Select the strongest colonies for trapping pollen, because weak colonies, you will be really doing a serious damage to them because they are struggling to raise their brood and you are depleting them from pollen. So make sure you select the strongest colonies possible with the large number of bees. And also timing is important. You can't just trap whenever you want. So look for timings when you see a lot of pollen foragers coming in. So that's what we do. We sit next to the entrance and see in a minute or 5 minutes how many pollen foragers are coming. And if you feel that's the strongest hive and that's the right time, then that's the time I want you to trap pollen. And the other tip is seal all entrances. I don't know if you keep auger holes like you see this hive on the left here. So we may have auger holes for aeration. So you have to, and bees may be using those as alternate entrances. So you may have to tape them all before you start trapping, because otherwise your trap will not be very successful. They'll start going from those holes rather than from your pollen trap. So what basically pollen traps do is they have this, like this is a simple one. That's a mesh they go through. So they're squeezing through and their hind legs have the pollen. The pollen drops because they're squeezing through those small entrances. Same thing with these, they have this mesh here. So bees have to go through the mesh, and the pollen loads, you see the yellow ones here, they drop onto the tray. So see that you have sealed all those before you start trapping. Install the traps and wait at least a day, because sometimes if you put this thing in, they may be confused. So I want you to put during the night, or maybe as soon as you think most of the bees are home. And the morning when they wake up, they go through this out. And then, so this is an insert. You see here the mesh here. So this comes off. So you can remove the insert for the first day or two so that the bees are directly flying in. And then you put the insert on so that now bees know how to get in, but they have to go through this mesh. So these are some tips. And so install the trap, as I said, a day before you start trapping. Just don't go directly and put this trap on, then the bees will be confused. And you may see some bees drift into the hive that is next door, or so you have to be you have to keep that in mind as well. So you have these, and then you can store this pollen for at least a year without any spoilage or degradation and protein supplement, the protein content. So use a Ziploc and collect that pollen. Don't trap more than 2 to 3 days. You may be hurting the hive if you trap for a long time. So those are some basic things. I think I am done here.
Ramesh, that was amazing. You know, I've seen a lot of talks, but I've never seen brain surgery on a honeybee. That was very, very cool. And you know, I'm familiar with the term endocrine for the human endocrine system, but exocrine, that's a new term for me. So we're all doing a lot of learning today. I wanted to ask you a quick question about one of your early slides. It looked like you did some analysis of Ultra B, and it looked like the Ultra B supplement contained no 24-methylene cholesterol. Is that true?
Yeah, for at least the one that we tested that here. Yeah, I don't know if things have changed now, but that was done 3 years ago, probably. Yeah.
Okay, that's interesting because Ultra B is something that we use quite a bit here in New Mexico. Okay, so we have a number of good questions. You know, New Mexico is not a huge agricultural state. We do, of course, grow chili peppers, which we're very proud of, pecans down south, alfalfa, and onions, but we're not a big ag state. So most of our beekeepers are sort of relying on native plants. So we had this question, have you analyzed the quality of native plant pollens? How do they compare to crop pollens?
Yeah, that's a great question. Unfortunately, I don't have an answer right now, but that's what the USDA grant that I mentioned. We want to collect a lot of different types of native plants as well. So our target is about 100 different species. So yeah, maybe if you email me after a year or year and a half, we may have some data at that time.
That's great. Okay, we have a question. If we happen to note larval mortality in our hives, How can we tell, or can we tell, if it's due to the absence of sterols?
Yeah, that's a great question, and I know you can really say that's because of sterols, because some of the starvation could be just because of the pollen dearth you have. There is no brood food in there, so yeah, that's a. I mean, yeah, eventually we may have to come up with some assay. There is no assay right now that can just say you give me some larva and I do an analysis and say or a quick field test. There is nothing like that at this point. So yeah, it's hard, at least to give you a short answer at this point. It's hard to say it just because of sterols. But again, if you have pollen, I don't know if the question is because specifically sterol, but what I would imagine, if you don't have pollen, then I have seen we have hives where we put in those flight cages that I showed you before, and if you have just protein supplement and the larva, you will not see much larva after a week because the endogenous sterols have gone because of the pollen. So as long as your pollen is there in the hive, I would assume there is adequate sterol in your hive as well.
Okay, great. I mean, I noticed that in our area, a lot of times, because pollen is coming in from different plants, especially during our honey flow, that we have this beautiful mosaic of pollen colors, so it's not uniform in color. We sometimes get purple, we get obviously different shades of yellow and orange. So we do tend to get a lot of diversity. Let's see. Anita has a question. Would you recommend probiotics to protect bees from pathogens and support their gut health?
Yeah. So again, I know I'm notorious running out of time, so I had slides on probiotics as well at the end. I didn't go over, but so probiotics, there is not much real evidence at this time on whether they are really doing anything beneficial to your bees. At least I have seen a couple of labs that are studying at least the beneficial effect, and I haven't seen anything really coming out from those at least showing. So I'm still a little careful suggesting any of the probiotics at this time because of lack of evidence. Maybe another couple of years you may see some published studies. And the other concern I have with probiotics is some of the bacteria, if you read the label, they have some bacteria. So there are some 8 core bacterial species that you're looking for that honeybees have, but they have some bacteria that are not in the honeybee gut in nature. And I don't know what those bacteria do. And the other thing you have to remember about probiotics is so these bacteria are surviving in the gut, right? They are in the more anaerobic. That means there is not much oxygen there. But many of the bacteria that I see in many of these probiotics are aerobic. That means they have cultured in the lab because that's easy to do in the lab. But what do those aerobic bacteria do in the gut? Are they doing nothing? Then that's a good thing. But if they are doing any negative things, then I think that's not good. So there is a lot to be known at this point. I know probiotics is a nice concept. I'm not discounting that, but I think you have to wait another couple of years to really get some good evidence.
That's great. Well, it certainly seems like you have a lot of work in front of you. I don't know how many postdocs you have, but you're going to need some more.
More
We
money
have
as
another
well.
question. We have another question. Would it be reasonable to add borage oil? I'm not sure if I'm pronouncing it right. Would it be
Borage, yeah.
reasonable
Mhm.
to add borage oil to a commercially available feed supplement like Ultra B?
Yeah, at least with the evidence I have, I think you can. But again, we haven't done any more targeted study on mixing borage oil into any of those. But as I showed you one of the study, and we have tested some borage oil as well. So we have seen a significant amount of 24 methylene cholesterol in the borage oil. So I think I would advise, but again, I don't have an exact quantity that I can suggest. Maybe use some small amounts, whatever you think is fine. And I think you can go with the borage oil.
Great. How about this question? How does bee bread compare with pollen for collecting and feeding?
So again, there is some, I think there was Kirk Anderson's work that has shown recently that they don't see much difference between bee bread versus the fresh pollen coming in. Again, that's only one study, so I can't really comment on that. But again, but if you look in the evolutionary process, I mean, the bee bread is, I think at least that study says the fermentation is only helping with storage, but it hasn't improved any value. The lactic acid fermentation, that the bee bread is basically your pollen that is converted into bee bread because of that lactic acid fermentation process. So that hasn't shown any evidence that it's increasing any nutritional value, but it just a shelf life is increased.
Great, thank you. Um, this question goes to the fact that we have a lot of folks that just mostly rely on flowers various native flowers. What are the best? Do you have any thoughts on what are the best flowers to plant with pollen that have sterol?
Yeah, great question. I know I anticipated this question, and I think I told them before that yeah, I hate to hit some list like 1, 2, 3, 4, 5, and so as I said, we have this USDA grant that we got recently, so we will be looking at, and I think then probably I can give you some good evidence that these are the top 10 plants that have the highest sterol or phytochemicals or amino acids or any of those things. But yeah, at this point, it's you can just go with whatever is attractive. That's what most of the Xerces Society or anyone that has a list is based on attractiveness of the flowers or the plant source to be. So yeah, again, I don't have much data on that yet.
No, that's great, but it's exciting to think that you're going to be doing some more of this research, and I'd like to think about inviting you down to New Mexico, and maybe we'll wait for a winter conference when we can show you that we actually have quite severe winters in the northern parts of the state. So
Oh, okay,
in
cool.
some ways, in Oregon, you're much more temperate, and
Yeah.
we do vary quite a bit from zone 4 to zone 9.
Yeah, looks like
So,
your New Mexico is more diverse. Yeah.
yeah, we have Africanized bees in the south, and we have pictures of hives with huge piles of snow on them in some of our more mountainous regions. So with that, I think we're going to want to thank you as collectively. We're so glad to have you here. As I mentioned with the other speakers, with you here physically. You're not here with us physically in New Mexico, but when we get to interact with you like this, we really feel that you're joining us here in New Mexico, and we're very grateful. We're grateful that the work that you've done. You've obviously chosen a career that means a lot to so many of us, and we appreciate your research. And we look forward. I think you're going to generate quite a few followers from the talks today. We've sometimes in the afternoon, and we noticed in the conferences, the population of attendees diminishes. But I'm very proud to say, and you should be proud to hear, that we still have as many people listening as we did first thing this morning. So thank you so much. We may get some questions that we can forward on to you from the chat box as a follow-up, because there were people that are interested in actually contributing to your research on native plants. And all of us want to hear more about the work you're doing on nutrition. So thank you very much. It's been
Yep.
quite a pleasure.
Yep. Thank you. Stay safe.